This video is membrane proteins 4. So typically the cell membrane is considered highly fluid. In two dimensions, basically in the plane of the membrane itself, and so that things can move. This is our cell membrane, laterally, so from one side to another, so basically in this horizontal plane. And so that's referred to as lateral diffusion. And basically, as you know, the cell membrane is made up of a variety of different components, not just phospholipids, but also proteins and other friends. And so these individual parts, each of the phospholipids, the protein, the other phospholipids here, each of them is kind of on their own journey as far as moving around independently. And the reason for this is that they are not covalently attached to each other. And it's a little hard to imagine from these kind of stoic looking figures in the textbooks, but there's some really great animations. For example, the inner life of the cell, which shows each of these little phospholipids and the proteins really floating around in space, kind of like a boat would float in a lake. And these individual parts can move around incredibly rapidly, and if there is random distribution of these individual components and there's no restrictions, then a membrane protein, for example, could completely move across the entire length of the cell membrane, that whole perimeter, within 40 minutes for a typical animal cell. And so that's a huge amount of movement. Remember, each of these items is individual, so that means this is a really random distribution of the membrane components, theoretically. So this basically is incredibly fast for how small these items are and how big a cell is. And so this provides a bit of a problem. So as you may recall, one of the functions of the cell membrane is as a meeting place. Basically that different enzymes in a biochemical pathway might all meet at the membrane. And so this doesn't really work. If everything's moving around independently at really fast rates, how can you possibly have a meeting? Like if you were going to have a meeting on campus with some of your fellow students and or faculty members, you have to pick a place and a time. And so basically there need to be some restrictions in many cases for cells. And so that's what this particular topic is all about. I have a little bit of an analogy, again, thinking about these individual components. So if you think of this analogy, and this is just to kind of help you think about the processes. I'm not going to ask you about this particular analogy on an exam ever or an assessment. So if you think about a lake or an ocean, and if you consider each of the boats to be proteins and the surface of the water to be the cell membrane, where we've got the surface of the water and then my very fancy boat here, then this boat is going to be able to float away randomly and be pretty hard to find unless it is anchored or tethered somehow. And so of course it could have an anchor at the bottom of the ocean, or if it were at a dock, it could be tied up. And so basically these are some of the ways that cells can organize different components, is through anchors or tethers or some other mechanisms. Another way that we can kind of subdivide a lake is using ropes and buoys. So sometimes there are buoys that are set up and then there's like a swimming area. This is a little hard to draw in without 3 dimensions. There might be a swimming area that's cordoned off using buoys and rope. And so basically the cell can use these sorts of different approaches to be able to restrict movement. And that way, that means that boats aren't going to go into the swimming area because that could be dangerous for those individuals. And also, when things are tethered, then we can know where to find them, or other people could know where to find them. So in a cell, the way that regions are subdivided for the membrane is through things that are called membrane domains. Note that this is not a protein domain, so we're not talking about a particular region of a protein that's folded in 3-dimensional structure. Instead, we're talking about, as a membrane domain, this is a functionally specialized region of a cell and this confines certain proteins to specific locations. And this ends up being incredibly important for a complex cell, like an intestinal cell, which we'll hear a lot about soon, to be able to have different functions on the inside of the intestine versus which might have to do with digestion and exchange of nutrients and waste, and another side of the intestine, which would be anchoring that tissue to the rest of the body. So there are 4 different ways that membrane protein location can be restricted, and so those are shown here. This is figure 11-31 in your book. And so basically, we're looking at this kind of funky little football-shaped cell, and it looks like it has laces, but those are actually some proteins. And so basically, through this figure, we see the 4 different ways that protein movement can be restricted in a cell. So in A, what we're seeing there is the protein, which is shown in pink, is tethered to the cell cortex. And so this means that it would be inside the cell. Another option to restrict location would be for a protein to be tethered to the extracellular matrix molecules outside the cell. So that's what it's showing here in yellow. A third option would be for proteins across 2 cells to be tethered to each other. So we see here these green proteins are somehow attaching to each other, and that's going to restrict some of the movement and mobility. And we'll hear more about this as different types of adhesion later. And then the one we're going to hear most about today is about diffusion barriers. And that's what these black rectangles are. And these will restrict proteins to a particular membrane domain. And basically what that means, think about this in 3-dimensional space. So this membrane is coming out towards us. This diffusion barrier would be the whole way around, kind of in this plane, in the front and in the back of the cell. And so here's one membrane domain, and that's shown with all these little blue proteins here that are located in that particular region. And then this would be the other membrane domain up at the what looks like the top of the cell, where there's some green proteins. And it's not a coincidence that all of the green proteins are up here and all the blue ones are here. Specifically, this diffusion barrier is blocking those membrane proteins from moving into that region. So basically, the blue proteins presumably can move however they would like, if you will, or however randomly they are going to move laterally through this part of the cell membrane, and likewise for the green ones. So they're not just like stuck in this exact spot, but they're going to move across this region. Wow, that's really hard to do. Okay. So now that you've seen a little bit about diffusion barriers, we're going to talk about a very specific setup of movement restrictions within epithelial cells of the intestine of animals. And basically, as you can see here, we have the black rectangles again, and so this is the specific name to the diffusion barrier. It's a tight junction. And the tight junction is made up of some specific junctional proteins, and basically they form an impervious seal across that site and across the two membrane domains. And there's more to their structure, which we'll hear about in the future. So the tight junctions are going to be found in this same location, kind of up towards the very top of the cells, and basically they're responsible for the orientation of these proteins. This would be the inside of the intestinal tract, or the gut. And so there could be things that are moving through this area, food and nutrients, and so then they might move through these proteins, for example, if those were channel proteins or transporters. Again, there's lots more about this that we'll hear more specifically later, but that's the basic concept here. This side of the cell is called the apical side of the cell. And then down here is the basal side of the cell, so basal-like base. And as you can see, the basal portion of the cell, which is in gray, so this is our cell, this is the basal portion down here. And apical is a spin-off of apex, which is the top or the tip. And notice that the basal side of the cell is located and attached to the basal lamina. And the basal lamina, which again is this yellow stuff, this is a mat of extracellular matrix which supports epithelial sheets of cells. And we're going to hear more about that later too, but for now that's the general setup. And basically, again, epithelial cells can be tightly linked together through these tight junctions. Epithelial cells are typically squamous or square-shaped, kind of this rectangular shape, and then they fit together in these sheets, and they all have this consistent shape, kind of like bricks, and they can form a very tight connection across them. I would call this a sheet. A couple other things to point out. So here the basal cell surfaces are down here at the base of the cell by the basal lamina, and then the lateral plasma membrane is along this way, and so this is in a perpendicular fashion to the other plasma membrane region, so the apical plasma membrane and the basal plasma membrane. And basically, as you can see, the tight junctions are going to restrict which proteins can diffuse into these different domains of the membrane. And just to be clear about the role of the proteins, the nutrients are coming into the cell through the apical side, and wastes are going to move out the basal and lateral sides. And this is due to proteins, which are located in each of those domains. Moreover, the tight junctions are, just as they say, they form a seal, so no proteins can diffuse across these locations, and so this means that those transporters allowing nutrients in or wastes out are uniquely localized to those membrane domains.